Hi everyone. So I'm going to talk about how we can use Bayesian parametric bootstrap for a model with intractable likelihood. So ABC again. So the structure of the talk will be, firstly I will explain a little bit about the stochastic models of collective cell spreading, and particularly for the cancer cell. And then I'm going to talk quickly on ABC. And in particular, we're looking at ABC important and rejection sampling and SMC ABC. And then next one will be the Bayesian parametric bootstrap. So just a bit background on the model and the context. So basically, collective cell spreadings has been very important for several processes. For example, wound healing and tumor growth. And it's involved a lot of mechanisms, but the main things we're looking at is the cell motility, cell proliferations of the individual cells within the populations. And so in mathematical biologies, they have been using several types of experiments, and one of them called barrier assays. So basically they put a whole bunch of cells inside the circular barriers, and then they put the nutritions and everything, and then remove the barriers and let the cells, and put in the incubator and let the cells moving outward. Whereas the squash assays is the opposite, where they put cells on the disc and then they use something to separate them out, and after that the cell were moving inward. So that's the difference. So for my models would be for the barrier assays where they move outward. So what we try to do is we try to estimate the cell motility and cell proliferations. So then we can compare, or we can come up with a systematic way to assess the effectiveness when we use different treatment or different doses of the treatment. So here is the example of the circular barrier assays for melanoma cells. So for the left image, where they apply a chemical to stop cell proliferations, so they only have cells motility. And on the right hand side, where they don't have the treatment, so you can see that the population is dense. It's much more than on the left. So the mathematical models. So there are two main approaches for the modeling for collective cell spreading. So the continuum models, where they use partial differential equations. So it's good in terms of the macro scales, but it's deterministic, and it doesn't provide insight into the uncertainty of the estimate. The other approach is the discrete stochastic models, or we call it the agent-based models, where we incorporate the random movement of individual cells, and we allow the cells heterogeneity as well, so it's really good, and they produce image-based data, which you will see a bit later. So it's very good, but it's a simulation models, and so the likelihood function are intractable, so therefore we have to use a likelihood-free method. So to simulate the experiment, we use the approximate versions of the exact continuous Gillespie algorithm. So I remember for the first work, we have compared the result when we use the continuous Gillespie algorithm and the approximations that what we use here, and the result is the same. But basically the continuous Gillespie, the continuous times Gillespie is much more computer expenses. So in the experiment, they only do the mono layer of cells. So therefore here we're only talking like two-dimensional models, so we don't do 3D models. So we use discrete unbiased random walk. So basically at each time point, when we let the cells move, they will allow to move either north, east, south, or west. We also incorporated the volume exclusions and realistic grounding effects. So it's basically if a locations, so if a cells attempt to move to a location, but there is already someone in there, so that move is aborted. So here are the notations. So the capital T we use for the experimental terminated times. Tau is the time step durations.
Brenda, you could mention that the fact you just observe this petri dish and then you stop the experiment, don't you? You stain it and then just take an image of that petri dish right at the end, say after 6 hours.
Yeah.
That's just one observation.
Yeah. Yeah, I think I have that a bit, 2 more slides.
Oh, sorry.
That's fine.
What is it for?
Yeah, yes. Yeah. So it's not the time series data. Yeah. So CT is the total number of agents. So for the model, we call that agents. For the real data, that's the cells. PM is the probability of an isolated cell's attempt to move a distant delta in the time interval tau. So by the isolated cells, I mean that a cells that doesn't have any occupied neighbor. So we're looking for 4 neighbors. And if that cells doesn't have anyone around, they have more chance to move compared to if they have some cells around there, because we have a characteristics or parameters that we're looking at called the cell-to-cell adhesion. So if the cells are close to each other, then they reduce the ability to move. And the distance delta, we chose that according to the dimension of the cells. So for the melanoma cells, the distance delta is about, I remember, like 23 micrometers. PP is the probability of a cell's attempt to give birth within the time interval tau. So to update from time t to time t plus tau, we sample CT agents from the CT agent populations and with displacement, and allow them to move with the probability PM times 1 minus Q to the 8. So basically, Q here is the cell-to-cell adhesion parameters, and this is from 0 up to 1. 8 is the number of the occupied nearest neighbors. So if the cell-to-cell adhesion is higher, then the ability to move is reduced. Or if 8 is large, say like 3 or 4, this probability becomes really small as well. So basically it couldn't move. Right, so this is how the simulation model looks like. So it's 2-dimensional, and I remember the dimension of the simulation is about 600 by 600. Sorry, I couldn't remember exactly the number actually, but it represents the dimension of the dish in the experiment. So here is the initial times where they all in the circle, and they was initially assigned equally around the space. And they move outward after 24 hours. So you will see a little bit moving out here, and after 48 hours and 72 hours. So I skip all of the image analysis step, but to identify the red line, the leading edge around here, I put the image into MATLAB image analysis, and so you will identify where the contrast. So for the real image, where the contrast between the cells and the background, and it's identify the location of the leading edge. And then we calculate the areas within the leading edge and convert that to equivalent circular diameter. So whatever we do for the experimental images, we do the same for simulated images. And the parameters we use for simulation or for stochastic models is PM PP and Q, but for mathematical biology, it makes more sense to convert them to cell motility rate, which is D, or cell diffusivity coefficients, D, and the cell proliferations, lambda. So basically, we perform stochastic ABC inference for PM, PP, and Q, and then convert that to the D and lambda and Q. Right, so the data available, we have, for the melanoma cells, we have data at 24 hours and 48 hours. We have 2 levels of initial number of cells, as Tony reminded before, so they are not series, not time series data, because when they take the image of the cells, they have to kill them first. So the 24 hours images are independent with the 48-hour images. So we have 2 scenarios with the treatment and without the treatment. So basically we have 2 by 2 by 2 experimental designs, and each of them will have 3 replicas. So we have the population scale image, where you can see the entire populations, as we saw a few slides before. And we also have the micro scales image, where we can see each of the cells. So after the image analysis, I get the summary statistics as here. So we have 3 radius of the expanding cells colonies, and we also have cell counts for 6 subregions and the proportion of isolated cells. So the image is here. Right. So the top here is the population scale image, and this one here is the individual scale image. So where we can see each of them and count them. So where do they take each of them? So they take it along a diameters, and these are the approximate locations. So when you zoom in, so the H is actually the zoom in from the G. And you can tell that the G is from simulated data. Right, so we have 15 summary statistics for each of the time point, each of the initial cells, and each of the scenario. And later on, I will explain a bit about how do we reduce the dimension, because 15 is still a bit high dimension for ABC. The blue dot here, so from H to I, what I try to do is, I try to map these cells' location to a 2-dimensional lattice. It's very close, but of course it's not exactly the same. And the blue cells are those considered isolated because they don't have any neighbor around. So here are the graphical summaries of the data. So you see that the top panel here, the cell count for 6 subregions, and the bottom panels is the isolated, the percentage or the proportions of the isolated cells. So with the treatment, with mitomycin C, with the treatment chemical, you see that the cells count are similar over time, which what we expected, because they don't generate anymore. Without the treatment, you see that the 48 hours, the number of cells count are much more compared to the initial and the 24 hours. And they are not so much differences across the location as well. So I think the experiment were really evenly distributed at the beginning. The percentage of isolated cells does not have much differences with the treatment, and without the treatment, there are more at the beginning, so at the initial time points, but later on, because there are more cells giving birth, so therefore less isolated cells. So the parameter of interest, we only have 3 parameters, and the question could be whether these parameters depend on the times, depend on the initial cells densities, or whether they are the same across two experimental scenarios, or whether we can estimate them from the summary statistics. Right, so now we move to ABC. Tony have talked a lot about ABC yesterday, so we may go through this one quicker. So basically, the goal of ABC is to search for parameter values that produce simulated data that close to the observed data. And I put up here some main steps of ABC for those who are new. So basically, the first step you do is sampling a proposed parameter theta star. Where do we sample that? It depends on the algorithm that you use. If you use ABC rejections, you sample from the bias or from the important distributions. If you use SMC ABC, then you sample from the previous iterations. So it's different depending on the algorithm. You simulate data, I call it Y-sim, from the models, given that parameter theta star. And then you compare the simulated data with the observed data by computing the distance rho. And in practice, we don't compare the whole data. Instead, we use the summary statistics. So the discrepancy function rho is for S, which is the simulated summary statistics, and S observed for the data, for the observed data. So basically, the nice things about ABC is that we can perform the inference just based on the summaries of the data, which is really nice in my case, because if you use the untitled data, you may have 20 or 20,000 or more individual cells. Whereas if you use the summary statistics, it's a much more smaller dimensions. Right, so that is the ABC posteriors. This is proportional to the bias, and this one is the approximate of the likelihood. And this depends very much on the epsilon and the summaries of statistics. So basically, in ABC, we introduce two sources of errors. The first one is when you reduce the whole data to the summary statistics. The second source of errors is when you approximate that when you introduce the epsilon. So the first source of errors have been discussed a lot in Plum 2010, where we need to be trace off between the information loss and the dimension reduction. The choice of epsilons is, if you have a lot of epsilons, then you compute quicker, but it is less accurate. And if you reduce the epsilon so small, then you have problem as well. So this one is the ABC important and rejection samplings from Fernhead and Frango 2012. So here when you start with a proportional density G theta, so given the data Y and the summary statistic S, so we call S observed, which is equal S of the data Y. So what we do is we simulate theta I from the proportional densities, and then you simulate the data set, you compute the summary statistics for that simulated data set, and then according to whether they are close with the observed summary statistics or not, then you give the weight, otherwise the weight will be 0. So the thing that I prefer is the SMC ABC rather than the ABC rejection. So the ABC rejection is parallel, but it's very slow, and it needs a lot of number of simulations. So SMC ABC has pioneered by Scott Sisson in 2007 in that paper, and until now, they have a lot of different versions of SMC ABC, but here I just give some summarize. So basically, it involves a sequential importance sampling techniques. So initially, you have n particles, theta I from 1 to n. You sample from the Brenda distributions, and then propagate it through a sequence of intermediate distributions to arrive at the target. So the target is according to the epsilon t. So SMC ABC aims to draw proposed parameters in sequentially higher posterior regions. So therefore, it improves and is much more better than the ABC rejections. However, it is good or not depend on, so the efficiency of SMC ABC depend on how far the prior with the posterior. If it is too far away, then you will have a lot of iterations, and it still have some disadvantages. So in this paper, we modify slightly the SMC ABC in Giovanni 2011 and Belmont 2009, which have some nice features that we automatically determine the epsilon t as in this paper, and we don't replace the good articles. So every time we move from one iteration to the next iterations, if the particle theta I that have generated the data set that closer to the observed data, then that would not be replaced by the next iterations. So the link to the detailed algorithms is in the paper. I'm not sure if it works. I hope so. Yep, so the detail, yeah, it's work. So the detail of the modified SMC ABC is algorithm 2. Here, if you want to look into details, each of the steps, and how we chose the weight and the resampling. Right, so that is the SMC ABC that we will use later on when we coupling Bayesian parametric bootstrap. So I said before that the more efficient SMC ABC, if we can find initial important distributions that closer to the posterior, so we reduce the number of the iterations. So that is why the idea comes from. So we will use the parametric bootstrap to generate the initial important distributions, and then use that in SMC ABC. But before we go to Bayesian parametric bootstraps, I will talk a bit about summary statistics that we adopt. They call semi-automated ABC from Fernhead and Frango 2012. So the idea is we perform a pilot ABC to find the regions of the non-negligible posterior. So in this pilot ABC, the epsilon can be large because we don't need to be more precise. We just need to get the areas where we know that the non-zero posterior densities. And then we draw m values of theta i's and the simulated data yi from that posterior regions. And you fit a multilinear regression models for each of the component of theta. And the explanatory variable here is the simulated data or the summary statistics of the simulated data. So after that, when we get the estimates alpha hat and beta hat, then we will substitute the observed data into that regressions, into that regression fitted regressions to obtain the estimate theta hat. So theta hat is just a point estimate, and we will use that as the observed summary statistics. So basically, doesn't matter how many dimensions you have at the initial here, and how many dimensions you have for the summary statistics in the regression models. The final summary statistics we use for ABC only have one statistic per parameters. So a bit later, for the melanoma cells example, we have 15 summary statistics, but we reduce that to 3 because we only have 3 parameters. So what is the Bayesian parametric bootstrap? So as Tony said before, Efron 2012 proposed the use of Bayesian parametric bootstrap and a reweighting scheme to approximate the posterior. However, in the approximations, so here's the idea. So here we assume that the summary statistic S is an estimator of theta. And to compute an estimate of theta, say here theta hat, and we just denote as equal to S observed to simplify the equations. So what we try is we generate B values of the simulated data given theta hat for each of the simulated summary statistics. Then, because here we can calculate the theta j, and then we calculated the weighting wj. So the sample theta j's, the weighted samples, theta j's and wj's, provide an estimate of the posterior distribution of theta given theta hat. However, the problem here is that you need to be able to calculate this. And for the problems where you have intractable likelihood, then this part is you cannot be evaluated. And therefore, the weight wj's cannot be found. For some special case, where the likelihood of S is symmetric, then the weight wj will be cancelled out, so it's only equal to the Brenda at theta equal to Sj. However, for many cases, if you don't have, if the likelihood is intractable, then you're not quite sure whether they are symmetric or not. So this one is just a very special case, and it's not in practice. Right, so the idea is we will use the weighted sample theta j and wj's to give the approximation to the posterior. But when we perform the Bayesian parametric bootstrap, we need the estimated theta hat. So where do we get that theta hat? It could be very time consuming if we run the whole ABC to get that theta hat. So the quicker way to do is we get the theta hat from the fitted regressions that we did when we performed the dimension reductions to figure out the summary statistics. So we use that theta hat from semi-automated ABC. When we get that theta hat, then we use it to simulate the bootstrap replicas and calculate the weight. So that's the idea. So here's the likelihood-free Bayesian parametric bootstrap algorithm. So we start with the observed data Y, the Brenda P theta, and how many bootstrap replicas that we want. So we perform the ABC pilot run to get the training regions for theta. Then we generate M simulated data set for the regressions. After we get the regressions. We fit the multiple linear regressions for each of the component of theta. And then we substitute the observed data to calculate the theta hat. And then use theta hat for the bootstrap procedure. So the bootstrap replicas theta j with the weighting wj will be used for the next step in ABC algorithm. So we use theta j and wj's to form the initial or the importance distributions in ABC, and I use that for both ABC important rejection samplings and SMC ABC. So we perform that for 2 test examples. The first one is the G and K quantile distributions, and the second one is the toggle switch models. But because of the time limit, I will skip the second one. So just the first G and K example. So the parameters for this model is A, B, G, and K, which corresponding to the locations, the scales, the skewness, the kurtosis. And for the simulated data set, we get this from these parameter values, and all using uniform Breuer. So for the regression procedure, we use 19, so we use 5% tiles of the data. So we have 19 summary statistics. After we fit the regressions, the point estimates we have is very close with the original value that we use. And because when we fit the regression, we check the adjusted R-square and it fits really well. So we consider that these summary statistics are highly informative for both 4 parameters. And the result is here, where the black line is the true posterior distributions. The red dash is the bootstrap, so the Bayesian parametric bootstrap. And you see that it's very close with the result in some cases. The bootstrap require very less numbers of simulations, and we have the result very close to the posterior, so that's what we want. We also use the bootstrap as important distributions for the ABC important samplings with ABC IS, and then the bootstrap for the SMC ABC, so the green and the blue. And the results that we save about 35% number of model simulations when we incorporate the bootstrap as important distributions. Right, so now come back to the melanoma example. So remember, for the initial summary statistics, we have 15, and after we perform regressions, we reduce to only 3 statistics for 3 parameters. And then we use that to conduct the Bayesian parametric bootstrap. So here's the result. So it's already converted to the biological parameters, D, Q, and lambda. So the red, the solid red, is after we use the parametric bootstrap in SMC ABC. And the black dash one is just the SMC ABC by itself. So they are very most likely the similar or the same. The blue is the bootstrap. So you see that in the Q and lambda, the bootstrap approximation by itself are very close to the posterior. For these, it's not really. Right, so that is the end of the talk. And here are the key reference.